So, hemoglobin estimation, we call it hemoglobinometry to measure hemoglobin level. So, there are different methods, but we will learn the easiest method, okay, and which is not followed nowadays in pathological laboratories. So, why do we learn it? Okay, there is a reason we should learn it. I will come to it. Okay, so hemoglobin estimation, right? How to measure hemoglobin level in your blood or in the blood sample given to you, right? So, the specific learning objective to describe the clinical importance of hemoglobin estimation, okay, recognize hemoglobin pipette and do pipetting in a single attempt, right? Determine hemoglobin level by Sahli's method, okay, remember and mind the spelling S-A-H-L-I, Sahli's method. It is called Sahli's method or Sahli's acid hematin method, right. Explain the absolute and relative hemoglobin scale, right. Next is interpret the result, list the advantage and disadvantage of Sahli's method. I just told that Sahli's method is an old method, okay, very primitive method of hemoglobin estimation and it is not accurate. Still we will learn it, okay. Right now if you have you seen in prescription doctor's advice blood for CBC means complete blood count, okay. Under complete blood count we have many parameters, okay. The parameters are hemoglobin, total count, differential count, then comes various indices like mean corpuscular volume, MCH, MCHC, packed cell volume, okay. So, these are the different and finally ESR. So, these and platelet count also. So, these are the things which are included in CBC, complete blood count. So, the very first parameter under CBC is hemoglobin, okay. And many a times doctors may also suggest hemoglobin separately, okay, hemoglobin estimation, okay. So, interpret the result, then list of advantage and disadvantages of Sahli's method and ultimately you will come to know that Sahli's method has got many disadvantages, right, and the hemoglobin estimation we will do by this method will not be accurate, still we will use it, okay. Enumerate other methods of hemoglobin estimation. আর কি methods আছে hemoglobin estimation করার জন্য and the most accurate method which is now being used in all pathological laboratories and we also use this in our central laboratory, you can visit and see, right, okay. And enumerate the precautions and source of error in this practical, क्या क्या error हो सकता है, right, okay. To determine the hemoglobin content of your own blood or of the blood given to you sample by Sahli's method. So, hemoglobin is converted into acid hematin. If we add acid in blood, hemoglobin will be converted into acid hematin. It is of brown color, deep brown color, right. So, hemoglobin is converted into acid hematin when we add any acid practically, but conventionally we use N/10 HCl. Okay, conventionally you know normal 1 normal HCl, okay, 1 normal means how much? 1 yes, 1 gram equivalent of HCl means how much gram? 36.5. Very nice, 36.5, okay. So, N/10 HCl which on dilution gives golden brown color. The color of the solution is matched with the standardized glass rod in the comparator. I am coming to it, okay. এখন হয়তো বোঝা যাচ্ছে না। So apparatus কি কি লাগবে এই প্র্যাকটিক্যালটা করার জন্য? ফার্স্ট Sahli's hemoglobinometer, right? So এই জিনিসটা আমাদের লাগবে Sahli's hemoglobinometer, okay, or it is also called hemometer, okay, it is actually hemoglobinometer, okay. So Sahli's hemoglobinometer আর এই বক্সের মধ্যে কি কি আছে, okay, see this, এই বক্সের মধ্যে কি কি আছে. So first a dropper, right, a dropper, N/10 HCl, First Sahli's hemoglobinometer, this one is Sahli's hemoglobinometer. Spirit, cotton and lancet, right, are a hemoglobinometer tube. Okay. So, here are the contents and also a stirrer, একটা glass rod থাকে, এটা পিপেট, গ্লাস রডটা আছে কোথাও? Yes, it's a stirrer, মানে মিক্স করার জন্য, right? Okay. See, hemoglobinometer comparator box, we call it comparator box. Now, this is the Sahli's tube or hemoglobinometer tube, Sahli's tube or hemoglobinometer tube, okay, this one, okay. So, one side in percentage and another side in gram per dl. So, mostly we use gram per dl, okay, we measure hemoglobin usually conventionally in gram per dl unless otherwise indicated. And this one hemoglobinometer pipette, this is hemoglobinometer pipette, okay, and here is marking up to 20 microliter. এটা 10 এটা 10 এর কত 10 এর আছে না 20 হ্যাঁ 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 10 এটা দুবার দিতে হবে। So 20 microliter, ওকে, এখানে একটা গ্র্যাজুয়েশন আছে, মার্ক আছে 20 মাইক্রোলিটার, ঠিক আছে, মানে 20 মাইক্রোলিটার অবধি আমাদের ব্লাড টানতে হয়, ঠিক আছে। একটা ব্লাড ড্রপ, গ্লাস স্লাইডে একটা ব্লাড ড্রপ নিয়ে এটার মধ্যে ক্যাপিলারি টিউব আছে, ঠিক আছে, তো এই ক্যাপিলারি টিউবটার মধ্যে আপ টু 20 মাইক্রোলিটার উই নিড টু ড্র ব্লাড, ওকে। এনিওয়ান হিন্দি স্পিকিং? অল আর বেঙ্গলি স্পিকিং? Okay, you are Hindi speaking. Okay, so you need to take a drop of blood on a glass slide and you need to draw blood with your mouth, okay, up to this point, the graduation. And there are some precautions, there should not be any bubble inside, okay, it should be a solid blood column. Glass stirrer. Now procedure. So first take n by 10 HCl in dropper and fill this hemoglobinometer tube up to the lowest point, okay, that is 2, okay, up to the lowest point, and we will keep it inside the comparator box. We will keep this inside the comparator box, okay. So this is comparator box. A comparator box ta carefully see this comparator box, okay. So in the middle there is a hollow, right, but on both the side we have a brown color. Can you see? On both the side we have brown color, right, on both side we have brown color, clear. The middle position is to keep this hemoglobinometer tube, okay. Okay. The thing is grossly আমি বলে দিচ্ছি, তারপরে আমি এটা পড়ছি, রাইট। তাহলে আমরা এখানে প্রথমে n by 10 HCl लेबो, ठीक है, हमें यहाँ पर n by 10 HCl लेना है up to mark 2, that is 2 gram per dl, ठीक है। This point up to the lowest point we need to take n by 10 HCl. Then we need to take blood here in this pipette up to 20 microliter, and we need to transfer this blood here, okay, we need to transfer this blood here. So what will happen? Blood is mixed in n by 10 HCl. We will keep it for 10 minutes so that acid hematin will form, okay. Hemoglobin will be broken down into acid hematin, okay. This acid hematin looks dark brown, okay, dark brown. Now if I put it here, so in between there will be dark brown and on both the side light brown, okay. Now I will add n by 10 HCl drop by drop and stir until a point where the color within the hemoglobinometer tube matches with the color on both sides. ठीक है. Now say for example कि हमने इतना तक n/10 HCl डाल दिया है और अब कलर मैच हो रहा है. Now I will check the reading. If it is 14, then hemoglobin is 14 gram per dl. ठीक है. Clear everyone? Grossly this is the procedure. Now read it out. So take n/10 HCl in dropper and fill the hemoglobinometer tube up to the lowest mark, that is 2 gram per dl. Keep the tube inside the comparator. Okay, now I will keep this inside the comparator. Under aseptic condition, prick your finger. Here you don't need to prick the finger because blood sample will be provided, right? Now wipe the first drop of blood because it contains tissue fluid. Okay, suck the blood in hemoglobinometer pipette up to 20 microliter. Okay, up to 20 microliter. Then clean the sides of the pipette. Okay, clean the side of the pipette and transfer the blood immediately into n/10 HCl, which is kept over here. Okay. Now rinse the hemoglobinometer pipette. ये आपको rinse करके clear करके रखना पड़ेगा and mix the content within the HB tube with the stirrer. Okay, the glass rod, right? So इससे आप mix करोगे and mix करने के बाद keep it for 10 minutes and let acid hematin form. Okay. Now dilute the solution in hemoglobinometer tube by adding drop by drop n/10 HCl and एक drop करके add करते रहो and mix करते रहो, right? And match with the standardized glass rod provided in comparator by lifting up the stirrer. Stirrer को ऊपर कर लो और इस ट्यूब को अंदर रख के दोनों साइड में जो कलर है उससे आप मैच करो कि इनसाइड जो मिडिल में जो कलर है वो मैच हुआ है कि नहीं, राइट? तो इट इज टोटली सब्जेक्टिव. ऐसा हो सकता है कि आपको लग रहा है कि मैच हो गया है लेकिन और किसी को लग रहा है कि थोड़ा सा लाइट है और थोड़ा सा डार्क है. ठीक है, ऐसा हो सकता है, राइट? एंड रिपीट द अबव स्टेप्स टिल द सॉल्यूशन कलर मैचेस द स्टैंडर्ड ग्लास रॉड कलर. ठीक है, एंड टेक द रीडिंग इन n/10 टेक द रीडिंग इन ग्राम पर डेसीलीटर by noting the lower meniscus. ठीक है, क्योंकि इसमें जो सरफेस रहेगा वो ऐसा रहेगा. ठीक है, with a concavity downwards, right? So lower meniscus आपको लेना पड़ेगा for reading. ठीक है. Now ideally the rule is you will do this experiment 3 times and you will take average of 3. Okay. But right now we will perform only one. Okay, only one experiment we will perform. Okay, now some theoretical question may be asked, like what is the oxygen carrying capacity of your blood? Maybe in your practical examination the question may come in this form कि आपको ये इंस्ट्रूमेंट दे दिया जाएगा. नहीं, आपको इंस्ट्रूमेंट दे दिया नहीं जाएगा because you need to collect this instrument from the table, right? So the question will be calculate your oxygen carrying capacity or calculate oxygen carrying capacity of the blood sample given to you. ठीक है, so you need to do hemoglobin estimation and you need to multiply your hemoglobin level with 1.34 because 1 gram per dl hemoglobin, 1 gram hemoglobin carries 1.34 ml, right? So if say for example your hemoglobin concentration is 14 gram per dl, so 14 into 1.34 is your oxygen carrying capacity. ठीक है, grossly, again grossly, because some amount of oxygen is also dissolved which we are neglecting. Okay, it depends on the partial pressure of oxygen, but grossly the oxygen carrying capacity will be hemoglobin percentage or hemoglobin concentration into 1.34 ml per 100 ml. Okay, so क्या क्या precaution लेना पड़ेगा? The blood column in the pipette should be free of air bubble. यहाँ पर आप जो blood draw करोगे, it should be free of air bubble. It should be a solid blood column, right? And for color matching, use the blank site of the hemoglobinometer tube. Okay, so one interference may be there. कि यहाँ पर graduations है, right? So ये graduation सामने नहीं आना चाहिए. So इसको आप ऐसे करोगे, ठीक है, ताकि graduation सामने ना आए, you can compare it carefully, right? Okay. Now glass rod should be lifted, जो stirrer है उसको ऊपर करके फिर compare करना पड़ेगा आपको, otherwise it will give you light color, and wipe off the excess blood sticking to the side of the pipette. Okay, transfer the content immediately. Okay, इसमें जो आप blood draw करोगे इसको immediately n/10 HCl में transfer करना है. Second is you need to wait for 10 minutes so that complete acid hematin is formed. Okay, and the procedure is to be completed without much delay because acid hematin को अगर आप ऐसे ही छोड़ देते हो तो वो धीरे-धीरे light हो जाता है, ठीक है? It fades with time. Okay, sources of error: not taking adequate blood up to 20 microliter, not giving enough time for formation of acid hematin, and using an old comparator box which is faded, right? And personal error, subjective error may be there, like आपको लग रहा है कि color match हो गया, किसी और को लग रहा है color match नहीं हुआ, ठीक है? ये subjective error हो सकता है. Okay, what are the advantage? ठीक है? So what is the cost? Very cheap. कुछ नहीं लगता इसमें, ठीक है? Very cheap, quick, and can be used for mass screening. This is important. This one your examiner will ask you कि इसका बहुत problem है, accurate नहीं है, ठीक है? There are various sources of errors, ठीक है? मान लो आपका hemoglobin बहुत accurate तो नहीं आएगा, मतलब 13.2, 13.4, 13.5, ठीक है? इतना आप नहीं कर सकते, आप बोल सकते हो 13 या 14, इतना बोल सकते हो आप इससे, ठीक है? लेकिन इन बिटवीन बहुत एक्यूरेट आप नहीं बता सकते इससे, ठीक है? एंड नंबर 1 इट इज सब्जेक्टिव, आपको लग रहा है मैच हो गया, किसी और को लगेगा कि मैच नहीं हुआ है, राइट? ओके, कैन बी यूज्ड फॉर मास स्कैनिंग. हियर इज द पॉइंट. तो लैब में हम इसको यूज नहीं करेंगे क्योंकि लैब में हमें एग्जैक्ट हीमोग्लोबिन कंसंट्रेशन चाहिए, ठीक है? एग्जैक्ट हीमोग्लोबिन कंसंट्रेशन हमें चाहिए. So exact hemoglobin concentration निकालने के लिए you need a different method, and that is modern method and much more scientific, ठीक है? And it is costly as well. So क्या method है? Spectrophotometric method. तो इसमें हम लोग क्या करते हैं? This is we are not going to learn because everything is machine, ठीक है? So nothing to do here in spectrophotometric method. Okay, this method we use in central lab where patient service is there. Okay, and this is for learning purpose only. Okay, not only learning purpose, we do it for mass screening. So what do you mean by mass screening? Say for example आप community health के purpose से आप लोग community medicine में visit कर रहे हो ना, rural health setup visit कर रहे हो ना, तो PHC primary health center visit कर रहे हो, तो say for example in a village you want to screen women of reproductive age group. Reproductive age group कितना होता है? 15 to 45, ठीक है? 15 to 45 is the reproductive age group. Okay, तो or maybe say for example you are for a project, it may be a UNICEF project or WHO project or some government project. You are to examine the hemoglobin concentration of all the adolescent females in a village of 5,000 population, ठीक है? So there हमें उतना accuracy नहीं चाहिए, ठीक है? आपको just पता करना है 13 या 14 या 15 या 9 और 10 और 6 और 7, ठीक है? So then we apply Sahli's method because एक दिन में हमें 5,000, 10,000 hemoglobin estimation करना है. Then we will follow Sahli's method, ठीक है? Clear? So here is the importance of Sahli's method for community purpose, for mass screening purpose. या फिर you are in a project where you need to estimate hemoglobin concentration of all the pregnant women in a village, okay, of 10,000 population, ठीक है? Then you need to do Sahli's method. But in laboratory where patients are admitted, for indoor patient, for any outdoor patient, OPD patients, we do this spectrophotometric method which is most accurate. You don't need to perform it in Physiology Practical, but your examiner will ask you what is the most accurate method and how it is done, okay? So one major problem in Sahli's method is that there are different variation of hemoglobin, right? Like sulfhemoglobin, methemoglobin, right? Okay, so these variants of hemoglobin, sulfhemoglobin, methemoglobin, they do not form acid hematin. So you underestimate. So whenever you do Sahli's method and आप hemoglobin concentration निकालते हो तो ये underestimation होता है, because you do not include sulfhemoglobin, methemoglobin, which are also minute variant of hemoglobin which is present in your blood. Okay, so here we use Drabkin reagent, okay, which is sodium bicarbonate plus potassium cyanide plus potassium ferricyanide. Okay, so here we use Drabkin's reagent instead of N/10 HCl, okay, and we form Cyanmethemoglobin. It is also called Cyanmethemoglobin method. यहाँ blood के साथ हम लोग Drabkin's reagent mix करते हैं, so that Cyanmethemoglobin is formed. Now this Cyanmethemoglobin, इसको हम लोग इसका optical density measure करते हैं with the help of light. Okay, the amount of light absorbed by the solution is measured by a photoelectric colorimeter provided with yellow green filters. Okay, so इसका optical density हम लोग measure करते हैं, and with that optical density we apply a formula and इससे हम लोग hemoglobin concentration निकाल सकते हैं. The details is not required for your level, okay, until and unless you are doing post graduation in pathology or something like that, right? Okay. So this is spectrophotometric method, and in all sophisticated labs we use this method. Okay, this is a Viva question for you in practical, right? But you need to perform this Sahli's method here. Okay, so as you know, for your hematology practical there are 10 marks, 5 marks is for your performance. Okay, and ये जो आप hemoglobin concentration निकालोगे इसका procedure, इसमें 5 marks है, and cross question में 5 marks रहेगा. Okay, cross questions are these. So what are the disadvantages of Sahli's method? What are the advantages of Sahli's method? What is the most accurate method? Right? So these are your cross questions. ठीक है? अच्छा, disadvantages की for Sahli's method, individual variation for color matching, जो मैंने बोला कि आपको लग रहा है कि मैच हो गया, किसी और को लग रहा है मैच नहीं हुआ, राइट? तो कलर ऑफ द स्टैंडर्ड्स फेड विथ टाइम, ठीक है, आप जितना छोड़ देते हो उसको कलर फेड हो जाता है. एंड नेक्स्ट इज सल्फ हीमोग्लोबिन, मेथ हीमोग्लोबिन, कार्बोक्सी हीमोग्लोबिन विल नॉट फॉर्म एसिड हेमाटिन. Okay, so you slightly underestimate, but that is not more than 0.2 or 0.3, right? Okay, so slightly you underestimate, but the advantage of Sahli's method is it is cheap. ठीक है, तो डेफिनेटली आप जब एक विलेज में 10,000 पॉपुलेशन को स्क्रीन करोगे, आप सायनमेथेमोग्लोबिन मेथड या कॉलोरीमेट्रिक मेथड, फोटोइलेक्ट्रिक मेथड आप नहीं करोगे, राइट? क्लियर? स्पेक्ट्रोफोटोमेट्रिक मेथड आप अप्लाई नहीं करोगे बिकॉज दीज आर कॉस्टली. ओके, so this is the thing. Now you need to start doing the practical, and another thing is that is slight modification, but this modification will not hamper your test. So initially we take N/10 HCl up to 0.2, okay, then in the pipette we take 20 microliter of blood and we mix the blood here, okay, then we add drop by drop N/10 HCl until matching, or we can also add drop by drop distilled water. That can also be done. There is no issues because acid hematin is already formed. So we can use either N/10 HCl or distilled water, no issues. Okay, we will use distilled water over here. Okay, till matching. So first take N/10 HCl up to 0.2, take blood up to this point, mix it and इसको आप छोड़ दो for 10 minutes. Okay, उसके बाद add drop by drop HCl until इसका कलर और ये दोनों साइड में जो कलर है इससे मैच होता है कि नहीं. ठीक है, मैचिंग करने के लिए आप पीछे एक प्लेन व्हाइट पेपर भी रख सकते हो. ठीक है, यहाँ पर ऑलरेडी दिया गया है. ठीक है.